Hello, everyone. Welcome back to the Beyond Clean Can't Touch This virtual conference, a full day dedicated to exploring the world of surface cleaning. I'm Adam Okada, your host for the day, and I'd like to once again take a moment to thank our event sponsor, 3M, for helping make this day of education possible. Dr. Michelle Alpha, sponsored by 3M, is a consultant at AlphaMed Consulting Limited. She is a board-certified clinical microbiologist and has her PhD in medical microbiology. Over the past 28 years, Dr. Alpha's research has been dedicated to hospital-acquired infections, specifically relating to monitoring, disinfection, and cleaning in the healthcare environment to reduce the risk of infection transmission. She's a well-known industry author, having published over 150 articles of study and research. Dr. Alpha has received many awards throughout her career, including the Champion of Infection Prevention and Control from IPAC. Dr. Alpha is going to share her insights on the importance of monitoring, cleaning, and disinfection of healthcare facility surfaces. She will outline the national guideline requirements for monitoring compliance of cleaning and disinfection to ensure staff are upholding such compliance standards. She will also discuss clinical studies that demonstrate the improvement in staff compliance when cleaning and disinfection practices are monitored. So I hope you have your disinfection wipes ready. I'm going to kick it off to Dr. Michelle Alpha.
Well, thanks so much, Adam. Appreciate the introduction. And I want to say how pleased I am to actually have the opportunity to participate in this series of presentations. So as has been mentioned, the topic I'm going to be addressing today is the importance of monitoring, cleaning, and disinfection of environmental surfaces in healthcare facilities. This is sponsored by 3M, and the disclaimer here, this is important information. My presentation will have data in it. It's based on published studies and current Canadian information regarding regulations, standards, guidelines, and practices as of June 11th, 2021. The requirements in other countries may be different, and Canadian guidance may change in the future. Always consult product instructions for use and follow the local regulations and laws. And this presentation contains an overview of general information and should not be relied upon in isolation to make specific decisions. So let's go on to the disclosures. I do consulting for a number of different companies, but I wanted to assure you the information presented today is my personal opinion and is independent of any company for whom I provide consulting services. And just to let you know, all publications that I mention will be listed at the end of the presentation. So my presentation is educational, and it provides the published scientific data related to monitoring of environmental cleaning and disinfection. And I personally am not endorsing any specific commercial product. So let's look at the objectives. The objectives of the presentation today are to review the role of environmental contamination in infection transmission. So within healthcare facilities, and that encompasses a broad range of different types of facilities, we want to look at whether or not the organisms, maybe they're multi-drug resistant, or maybe they're just pathogens potentially there that could infect patients. If they're in the environment, what role does that play in other people getting those infections? And the second objective is to describe the value of monitoring, cleaning, compliance. And the third one is to discuss the published data on cleanliness monitoring using ATP and UV marker removal. Okay, it's an exciting session today because we actually have poll questions. So this is the first poll question, and it's basically what type of facility are you currently working in? And if you could take your cursor and go up to the little circle that represents the facility that you're working in, that would be great. And once I've I'll give you 10 to 15 seconds, and then what we'll do is kind of review who's in the audience in terms of the mix of people that are participating in this particular webinar. Okay, so I think I'll click on. Oh, look at this. The vast majority of attendees are in hospital environment. I'm really happy to see that there are some people from long-term care, and we also have another large group from ambulatory care. No research or academic participation today, but we do have industry related to healthcare. So this is a great mix of individuals, and I think it's important to recognize that monitoring of the environmental cleaning and disinfection applies to all of these types of facilities. So that's the type of poll questions we're going to go through. And after each section, in terms of the objectives, I will activate a poll question, and it will be similar to what you've just experienced. So great practice, 100% on your participation in the poll questions. So let's look at objective 1. I want to review the role of environmental contamination in infection transmission. So the first thing that we always tend to look at is, what do the guidelines say? And in this slide, you'll see I've listed the PICNET guideline from British Columbia in Canada from 2016, the PIDAC document from Ontario that was last updated in 2018. And I want to bring your attention to the fact that there is a new Cleaning and Disinfection of Healthcare Facilities CSA document that was published or put out in 2020, and it's an excellent document that is one of the most current ones in Canada at this point. In addition, there is the CDC guideline as well, and particularly I will be referring to annexes B, C, and D. And if you look at all of these guidelines, every one of them have good reviews on the published data that evaluates the role of environmental presence of microorganisms on the role of the environment in transmitting these to subsequent patients. And all of them confirm the role of the environment in infection transmission. Now, I myself have actually been very much convinced of this by the particular studies, and I've listed some references at the bottom of this slide. But in terms of the evidence of hospital-acquired infection, or HAI, transmission related to the environment, specifically for pathogens such as MRSA, vancomycin-resistant enterococci, VRE, or C. difficile, or C. Clostridioides difficile, there's enough data in the literature to convince us that, sadly, patients admitted to a room previously occupied by a patient with MRSA, VRE, or C. difficile have significantly higher risk of infection or colonization with these same organisms. So it's unfortunate, but the reality is that this attests to the importance of the EVS role, or the environmental services role, in ensuring that the healthcare environment isn't acting as a reservoir of these types of pathogens and putting our patients at risk. Now, there was a very interesting publication that came out in 2019, and this is the data that I'm showing you here. And what they wanted to look at was the presence of multi-drug resistant organisms. And in this case, the multi-drug resistant organisms that they evaluated included MRSA, VRE, or resistant gram-negative bacilli. And what they did was, there were 399 patients that were admitted to a healthcare facility and gave consent to participate in this study. And their consent was acquired within 24 hours of admission. And it was also evaluated during the hospital stay. So these individuals that were admitted gave consent to have evaluation or samples taken at enrollment and during their hospital stay. And this was done in 2 hospitals in Michigan. So you can see here that the graph on the left is showing the patient hand cultures that were taken. And you can see that on admission, there were 10% of people coming into the facilities that actually had one or some of these multi-drug resistance on their hands at the time of admission. And you can also see the percent on the left-hand side, which is the percent of having any of these organisms on their hands. And you will see that in the environment, there was about 28% that actually had these multi-drug resistance in the environment. Now, I don't know about you, but it was disconcerting to me to see that 28% of the environment had multi-drug resistant organisms. But keeping in mind that the samples were taken within 24 hours of admission, what the studies did in addition was they also looked at what happened for those patients that were admitted within 0 to 8 hours of admission. And so this yellow bar that I'm showing you indicates that if the patient was enrolled within 8 hours of admission, 8.1% of them had multi-drug resistant organisms on their hand, and the room only had, other rooms, not only, but the rooms had 5.4% of these multi-drug resistant organisms. So you can see that by monitoring what was on the patient hands using imprints on agar plates, and also by taking samples from the environment to assess for these specific organisms, it's clear that both the incoming patients and their rooms actually can have multi-drug resistant already on them. And the other component of it is that the patients, when they come in, if they don't have organisms on their hands and there are organisms in the room, what is the likelihood that they can actually acquire them? So this study also looked at the new multi-drug resistant organism acquisition rate for patients. And they found that people coming in that did not have organisms on their hands, there was 24.6% of cases where they acquired a new MDR per 1,000 patient days. And they basically did molecular typing and showed that the molecular typing confirmed that the same multi-drug resistant organism was on their hands as that was found in the environment. And you can see down here, the blue bar represents new acquisition. So in terms of the percentage that acquired a new MDRO, you can see that on patient hands, there was 6.2% of people that acquired a new MDRO, and there was 21% of the environments that didn't have an MDRO in them that acquired a new one. And so what it shows you is that there is rapid environmental contamination from newly admitted patients, and some of the rooms have MDROs already in them before the next patient is admitted. So there's two take-home messages here. One is that incoming patients rapidly contaminate their environment with the same MDRO they have on their hands. And the second take-home message is that in some instances, the rooms in healthcare facilities actually do have organisms in them, and that this can lead to new acquisition of MDRO by the patients that are admitted to these rooms. Now, the other interesting thing about this study was that there was two hospitals, we'll call them Hospital A and Hospital B. And I'd like you to notice that the resistant organisms that were evaluated were MRSA, resistant gram-negative bacilli, and VRE, shown on the left-hand column. And I guess the incidence of these MDROs varies by healthcare site. It was interesting to see that Hospital A, the biggest issue they had was with the environmental presence of VRE. You can see that 46.1% per 1,000 patient days was their incident of this type of pathogen. So it was the predominant concerning organism in Hospital A. Whereas if you look at Hospital B, you will see that the resistant gram-negative bacilli were 53.3% in terms of incidence per 1,000 patient days. So Hospital B had a different set of problems, if you will, in terms of the resident environmental organisms playing a role in transmission to patients. And what was interesting to me was that Hospital A was using a cleaning and disinfecting agent that included hydrogen peroxide and peracetic acid, whereas Hospital B was using a quaternary ammonium agent. Now, this is not to say that one is better than the other or one is worse than the other, but just to indicate that in some instances, there may be differences in the key pathogens that are causing problems in a healthcare facility, and sometimes that may be related to the type of cleaning and disinfectant agents that are used. And on that note, I'd like to go to poll question number 2. And this poll question is, which of the following organisms is currently the most common hospital-acquired infection in your healthcare facility? And again, you can think about it. Don't worry about you don't have the data. In your perception, which of these organisms is the one that you perceive as causing the most angst in terms of hospital-acquired infections? And if you can go ahead and click on the circle that best represents what is happening in your facility, just so we get a sense of who in the audience is having which type of multi-drug resistant organism problems. And overwhelmingly, you can now see that the MRSA situation is one that is very, very troublesome, at 60% perceiving that this is the biggest hospital-acquired infection problem they have. And the second most common one is C. difficile. And you can see also there is quite a few multi-drug-resistant gram-negative rod bacilli. So we have 20-20 split between C. difficile and multi-drug-resistant gram-negative bacilli. And we have overwhelmingly 60% of the facility or the participants perceive MRSA as their biggest problem. And again, the reason I raise this is that every facility may have a different set of problems, but the bottom line really is that the presence of these organisms in the environment can be driving or leading to the predominance of some of these organisms in terms of transmission. Now, I would be the first to say the environmental reservoir is one factor, but there are many other factors, obviously, in terms of the cleaning and disinfection methods that are being used, in terms of the approach taken for healthcare providers, in terms of their hand hygiene, et cetera. So the reservoir is one of these factors, but it's important to recognize that it is one that we can actually control and do something about. So let's move on to objective number 2, and that's to describe the value of monitoring cleaning compliance. And I would like to kind of preface this discussion by talking about, okay, what is environmental cleaning disinfection anyway? And you'll notice I put cleaning/disinfection. It's not just cleaning, but the concept of also disinfecting those surfaces so that if the cleaning, and cleaning can never remove all the microbes from the surface, so if the cleaning, the physical action doesn't remove it, that we have a disinfection component that will eradicate or kill anything that remains after the physical cleaning. So what is environmental cleaning? Cleaning is using a detergent with physical action. Good old elbow grease. And by using a detergent that facilitates removal, you're going to be removing any of the debris that might be present on the environmental surfaces, organic material that might be there, including patient secretions, and you will remove to some degree the microorganisms. But in addition to the physical action, we need killing action, and that would be the disinfection aspect. So the killing action is exposure to an agent that kills microorganisms. Exposure of the environmental surfaces, such as toilet facilities, such as bedrooms, bed rails, all the high-touch areas in a patient care environment. And the disinfectant may be chemical in terms of a liquid, a vapor, or a gas. It could also be a UV light system that is used for whole room decontamination, steam, etc. And the other component is there are some products that are one-step cleaner disinfectors. So the agent itself has the ability to properly clean, and in addition, it has chemical liquid disinfectants in it that can actually kill microorganisms. And if you're relying on a liquid chemical in order to do the disinfecting, it's important to ensure that you check the manufacturer's instructions for how long the contact time is in order to get the efficacy that the manufacturer can state is going to be given by that product. If the manufacturer's label says the surface must be wet for 10 minutes, that it's, you have to be very cautious about that, because most times when a surface is wetted and wiped with a wipe or with a cloth, it will only remain wet for anywhere up to 3 minutes or less, depending if it has alcohol in it or not. So it's important that in order to get the efficacy you believe you're getting from the product, you need to make sure that the surface remains wet for the contact period that is indicated by the manufacturer of that product. Okay, so with this in mind, in terms of environmental cleaning and disinfection, remember it can be a two-step process where you do cleaning, which is physical action, and then you do a second step, which is disinfection. And one of the things that is becoming more commonly used these days are the no-touch room disinfection methodologies. So that could be fogging using vaporized hydrogen peroxide, VHP, ozone gas, superoxidized water. It could be UV irradiation, the nice robots that put UV irradiation into the room, which eradicates microorganisms on surfaces, or it could be steam. But it's important to remember that even if these no-touch room disinfection methodologies are used, you still must do the physical cleaning, and you still must monitor to ensure compliance with the physical cleaning. I have a colleague who introduced no room touch disinfection, and in that person's case, it was UV irradiation. And it was interesting. They found there was a lot of failures. And when they went back to identify why this was happening, it was happening because the EVS personnel felt, well, you have this fancy new machine that can completely kill any organism in the room. And, the cleaning I'm doing, I can speed things up and get things done quicker because I don't need to do the cleaning in the way that we normally did it. And it was very clear when they started monitoring cleaning compliance that it wasn't being done appropriately. And that was the reason why the UV radiation, after its treatment, there were still organisms being detected. And this was in a study that was being done. But the point here is that even if you use no-touch room disinfection, you must still do the cleaning, the physical aspect of it, and that it's important to monitor compliance to identify if there's any issues still being done with the cleaning, because if there is, the no-touch room disinfection technologies will not be able to function properly. So looking at cleaning monitoring and looking at the guidelines, this is the new CSA document, and it clearly states cleaning effectiveness audit. A cleaning effectiveness audit and its frequency shall be performed in accordance with the healthcare facility and the environmental services policies and SOPs. And this is the first document that has come out with a shall regarding monitoring of cleaning compliance. Shall means it's a regulation. It's not an option. It's something that is expected and should be done. And furthermore, you see it states that it shall be measurable. It shall include at least a visual inspection and one other audit type. And it shall provide a measure that can serve as a quality indicator, and it shall establish benchmarks for acceptable cleaning performance. Now, this is a Canadian guideline and obviously applies to Canadian facilities that are healthcare facilities because this is our national guidance document. But I also want to mention that the CDC guideline, which was effective as of 2019, states all hospitals are encouraged to develop programs to optimize the thoroughness of high-touch surface cleaning as part of terminal room cleaning at the time of discharge or transfer of patients. So even though it may not be a regulatory requirement in the United States, it certainly is a recommendation and certainly is encouraged. So in both countries, this is something that is seen in the guidelines as a useful thing. So the CSA guidance documents identifies key performance indicators, and they include visual inspection plus one of the following parameters. And in today's session, what we're going to focus on is marker removal and ATP testing. The other key performance indicators include culture and survey, and these are important ones as well under certain circumstances, but today's focus will be on marker removal and ATP testing. So let's have a quick look at what does this mean. Now, remember, there's multiple manufacturers of commercial kits for ATP testing, and I am not promoting any particular one, but the ATP test monitors the level of organic residues on the surface after cleaning and disinfection. And it's important to recognize that even food residues contain ATP. And certainly, it's important to understand that ATP is not solely testing for the level of microbes on the surface. It's testing for any organic material that contains ATP. And it cannot detect low levels of microorganisms. So even if you pass your ATP test, you must keep in mind that doesn't mean there's no organisms on that surface. It simply means that the ATP levels from organic residues are low enough that it shows that the surface has been adequately cleaned. So for ATP testing, the methodology that's used is there is a test tube with a swab in it. It's used to swab the surface of interest, usually or always high-touch surfaces. And then what happens is that swab is inserted back into the self-carrying tube, and the tube is mixed and then inserted into a handheld luminometer, this little device that you see here. And in a few seconds, it will generate relative light units. Relative light units are a measure of how much ATP is present in the sample. It's a very rapid process, and it's very easy to do. It's quantitative, and it's a one-step process, which is wonderful. But it's important to remember standardization is really important. Remember that different kits from different manufacturers have differing sensitivity to detect ATP, and as such, the relative light unit, that's the cutoff for clean versus not clean, differs between different types of kits. And in terms of standardization within your facility, whatever kit maybe you choose to use if you're using ATP, it's important to standardize the high-touch sites that will be tested and what surface area to swab. Remember, it doesn't help you at all if in one instance a staff person swabs a 2 centimeter square area on an item, and then the next time, the next day, the next person swabs a 5 centimeter square surface area. It doesn't mean that, because that means you cannot compare those results from day to day. So sites need to standardize which high-touch surfaces will be tested, and also for those sites, what surface areas to be swabbed, and then they have to establish or determine what relative light unit, RLU cutoff, will be used to say, okay, that surface has been adequately cleaned and then, or not adequately cleaned. So that's describing the ATP testing. If we look at UV visible marker, or any type of marker for that matter that is used for this purposes, and again, there's multiple manufacturers to monitor this to determine if cleaning has been done. Remember, this type of marker is a 2-step process, and I'll explain that in a minute, but it monitors if the surface was wiped. The marker is very easy to remove, and as long as the surface has been somewhat wiped, that marker should be removed. So the 2-step process is, number one, staff go around and they apply UV marker to the high-touch surfaces that they want to test. And again, they need to standardize which of those high-touch surfaces will be evaluated. And then after cleaning, either the next day or a defined period after the marker was applied, they come back and use a handheld UV light to assess whether or not the marker was removed. And in the pictures I'm showing you here, the UV visible marker will fluoresce under UV light, and you can see it in the left side picture at the bottom. And if that marker is removed, you will see there's no fluorescence as shown in the picture on the right. So it's a 2-step process, pretty straightforward. Has the marker been removed or not? And it's documented as, in most instances, as the marker was completely removed or it wasn't completely removed. Okay. So let's look at the guideline. This is the CSA guideline, and they actually do a very good job of comparing the fluorescent marker to ATP. And you can see that the description is that the surface is marked with an invisible agent, and you evaluate it after cleaning and disinfection using a revealing agent to see if the mark was removed. Whereas for ATP, the surface is tested after cleaning and disinfection to determine quantitative level of ATP that remains and assess whether or not that is appropriate or not. The element evaluated for the fluorescent marker is mechanical action. The element evaluated for ATP testing is whether there is organic residue still remaining. The pros are, for both of them, they're quick feedback. The difference between a 2-step process and a 1-step process is that the ATP result gives you quick results because you go in, after cleaning, you do the testing, the results are available, whereas in a 2-step process, you have to do 2 steps to actually get that information. They're both easy to implement. The results of the fluorescent marker are easily understood, and it allows direct assessment of cleaning and disinfection thoroughness. For ATP, it's a quantitative result and the assessment of organic residues after cleaning and disinfection. The cons in this testing are that fluorescent marker is not an evaluation of a disinfection capability. It doesn't directly measure microbial contamination, and remember that surface texture might affect the removal of the marker. For ATP, remember the absence of organic matter doesn't rule out the presence of microbes, and it can't compare results across system So, for instance, from one facility to another, if you're using a different area of sampling and you don't have it standardized, if it's not standardized, you can't compare the results. But in a facility, from time to time to time, with the standardized approach, you definitely can monitor and show trends in terms of the efficacy of monitoring of cleaning. And there's a potential of certain environmental surface disinfectants to interfere with the ATP system, which can give false or positive results. But as long as you take that into consideration, it should not be an issue. So interpretation for both of these requires a predetermined acceptable residual fluorescence on the surface for the fluorescent marker. And in most cases, people will use complete removal versus incomplete removal or no removal. For ATP, a maximum level should be established with a background or another method so that for the facility, they know that above a certain RLU, that surface needs to be recleaned. Below that RLU, it's okay. And the same for the fluorescent marker. If it passes, the surface doesn't need to be recleaned. If it doesn't pass, that surface needs to be recleaned and disinfected. All right, this brings us to our next poll question. So get your trigger fingers going. And in this particular question, it is, what cleaning and disinfection monitoring method is routinely used in your facility? So you have a choice. Do you use ATP plus visual inspection, or do you use UV and visual inspection? Do you do culture of the surface and visual inspection? Do you use visual inspection only? Or are you not doing any monitoring, so none of the above? Okay, it's always very exciting to see the results of these polls. So let's go ahead and have a look at where we are in terms of this aspect. So excellent. Look at this. We have 50% use ATP plus visual inspection. In this group, there aren't people that are using the UV marker with visual inspection, and no one's using culture and visual inspection. And visual inspection alone is used in 31%, and none of the above in 18.8%. This is a wonderful example, and in many cases, I think it raises the issue in terms of what is kind of the recommendations. So in Canada, using visual inspection plus at least one of the markers would be considered to be a requirement. So visual inspection alone would not be seen as being adequate, and none of the above would not be seen as adequate. And I appreciate the honesty of the participants for letting us see this data, because I think it's important to understand what the current situation is. And there is no wrong answer in this. This is just understanding what's going on. Kudos to those who are using cleaning and monitoring. And for those who aren't, consideration to the value of it. And I'm looking at Objective 3 to help us identify what is the value of doing the cleaning monitoring. So I wanted to discuss the data on cleanliness monitoring using ATP and UV visible marker. And what I want to share with you is that when we actually did a year-long clinical study to assess the importance of this. But before we did that, we undertook a research study, which was published in 2013. And Adriana Treisman was the graduate student that was involved in this study. And the first thing we did was we obviously got approval from the unions for the EVS workers. And the EVS workers knew that the study was going to be undertaken, and they were quite okay with it. And before we actually started the actual study research part of it, we wanted to know the background cleaning monitoring facility. So you'll see on the bottom axis, we did 4 consecutive weeks where we used a UV marker, which was the 2-step process I described, and we assessed what was the percent cleaning compliance, and the percentage is shown on the y-axis here. The dark bar in week 1 was before we, how will I put it? The EVS staff knew we were doing this, but we hadn't started giving any feedback or anything along those lines. And we found that there was around 50% or thereabouts cleaning compliance. That means that 50% of the sites we assessed had not had the cleaning marker removed, the UV visible marker removed. And as week one was when we actually kind of started before we were giving feedback, but we didn't start the study for the first few weeks, even though the EVS staff knew that we were undertaking a study in all 4 weeks. And you can see that over that 4-week period, the cleaning compliance hovered around 50%. And this is not unusual. If you look at the published data, this is our study or our experience in our healthcare facility. But if you look at the research data, you will see this is pretty much around what the percentage cleaning compliance is when no monitoring and no feedback is being done. Now, it was interesting because we had a meeting before this data was generated, and that's why you see that line, that dark line at the top there at 90%. We met with our EVS staff and we asked them for their feedback. We explained the study and why we're doing it, and it's not punitive. This is just so that, we can actually monitor and improve things, and that if there are issues, you have a way of being able to show that, hey, it's not the EVS people that are the problem because we are cleaning at an acceptable cleaning rate. It has to be some other issue if there's an outbreak or something like that. And we asked them, what % cleaning compliance do you think you should achieve on a regular basis? And they actually said to us, 100%. Absolutely. We know we're doing a good job. We can clean to 100%. And so I asked them, I said, okay, I trust your confidence in it, but I would suggest that you give yourself a little buffer room. And so we settled on 90%. That was what they felt they could achieve. And then we started the study where we actually did the monitoring, and it was done on a weekly basis. There were a number of rooms done every week and a fair number of rooms on every ward. And what we did was, you can see here, over the 24-week period shown on the bottom of this graph, you can see at the very beginning, even though they had been at around 50%, once the study started, we were hovering at about 60 to 70% in the first couple of weeks. But look at the jump. They very quickly, once they started getting feedback on a weekly basis, and it was a team approach, it was identified to the team that this area hadn't been properly cleaned, and the feedback for that, the team then puts peer pressure, and there was pride in making sure that the team was above 90%. And you can see that with ongoing feedback, and this feedback was given on a weekly basis, the overall compliance with cleaning went from about 50% before this study, and after 24 weeks on the wards that were given continuous feedback, they were achieving 90% on a very regular basis. Now, one of the things that we learned from this study when we analyzed the data on a more detailed basis, training of new environmental services staff is truly important. That when we looked at the data and we could drill down to individuals, although it was not fed back to individuals, it was fed back to groups, we found that if staff who trained the new staff, new EVS staff, had low cleaning compliance, they trained the new staff to the same low compliance. Whereas if the staff training had higher cleaning compliance themselves, they trained new staff to very high compliance levels. And so you can see, and we took this as a very important take-home message, that really it is important to have dedicated training done when new EVS staff start. They are the front line in infection control in healthcare facilities, and we really need to ensure they're properly trained and ensure they have proper protocols and that there is feedback given to them so that they maintain their competence. So these were a take-home message that came through to us. And Jackie mentioned in her presentation, it's important to know who's responsible for cleaning what items. And I have a little short story to tell you because as a separate part of some of the research that we were doing, we were marking the commodes that patients use, those mobile toilets that are notorious in healthcare facilities. And what we found out was my research tech came back and she said, Dr. Alpha, I don't need a UV marker to tell me anything. There is actual physical poop I can see on some of these commodes. I know nobody's cleaning them. And indeed, that was the case. We talked to environmental services and they believed it was nursing that were cleaning them. And the nursing staff believed that the EVS people were cleaning them. So it was true. My research tech was right. Nobody was cleaning the commodes, and these commodes move from room to room. So I think it's the other important part about training is making sure it's clearly defined who cleans and disinfects what items in each room. Okay, the other study that we undertook, and this was an important one, it's a question of, if you do this and you are monitoring cleaning, can it actually have an impact on reducing hospital-acquired infections? And so this is a study that we undertook. It was the year-long study. We went from November 1 to October 31st on each of the years that I'm showing you here. And you can see the cases per 10,000 patient days on the y-axis. So in this case, we're looking at C. difficile. The Canadian national standard for C. difficile cases is 6.04 cases per 10,000 patient days, and the red arrow shows that. So you can see that the blue bars represent any cleaning compliance, just all comers. What was the background rate of C. difficile? And you can see from 2009 to 10 over to 2012 that there had been a gradual decline up until 2011, 2012. The study year was 2012 to 13. And you can see that when we had any cleaning compliance, it had gone down, it had trended down. But when we were looking at those wards that achieved greater than 80% cleaning compliance, the red bar, it was significantly lower, even compared to the blue bar in that year for any cleaning, and compared statistically even to the downward trend, it was statistically lower. There was less cases of C. difficile that year than there had been in the previous years where monitoring was done. And our infection prevention and control staff told us they really noticed that because the isolation carts that were needed, there was far fewer of them needed out on the wards. And the same data was found for both MRSA and VRE, that there was significant reduction in these cases when 80% cleaning and disinfection was achieved. So there is some data to support the idea that it is important to monitor cleaning so that you can get people's compliance up to a good level, and that will reduce the environmental reservoir. Now, just to show you a more recent study, this is a 2020 study that was done, and they looked at ATP and culture, and they looked at surface, the environmental surfaces, and hand contamination. And I like this study because it used a very standardized approach to what they were looking at. So first of all, they defined the surface area. They did a 4 by 4 centimeter surface area testing for both the ATP and for the aerobic cell count, or ACC. And in that aerobic cell count, they were looking at predominantly, they were monitoring things like MRSA in particular, and the standardization of the surface area for both of these was important. The clean cutoff, in this case, they were using an ATP testing kit, and the cutoff for it was established at less than 100 RLUs. So if it was less than 100 RLUs, that meant that the cleaning had been and disinfection had been done appropriately. They also defined a cutoff for the colony count, or CFU, less than 2.5 CFU per centimeter squared. And they also wanted to assess whether there was any MRSA on hand culture. And hand culture was where they put on a sterile glove, they moistened it with sterile water, and then that person touched all 5 high-touch sites that they were being evaluated, including the phone, the call button, bed rail, table, and toilet rail. And then after they touched all those surfaces, they pressed it to an agar plate that was selective and allowed them to detect methicillin-resistant Staph aureus, or MRSA. So now on the data at baseline, you can see the dark bar, the ATP assessment, is 43%. That's the percent contamination. So remember, this is the reverse of what I was showing you before. This shows what percentage were still contaminated after the cleaning and disinfection process had been done. And at baseline in this center, 43%. So that means their compliance was about 57%. So similar to how our baseline was in our study. And that the clean off for the colony count, the gray bar, was the percent contamination was 50%. And there was 7% contamination in this hatched bar, which is the presence of MRSA that could be picked up by your hand if you touch the environment. The next stage was to then provide education and start giving feedback. And you can see there's a drop in the percent contamination. The dark bar, the ATP testing, dropped down to 30% that still were contaminated after cleaning and disinfection. The total colony count, the ACC gray bar, dropped as well from 50% to 18%, and the MRSA hand contamination dropped to 2%. What was interesting in this study is they then went on to introduce whole room decontamination with UV radiation. And you can see here that compliance with the ATP component of it dropped down so that there's only 9% contaminated that didn't pass the cleaning monitoring with ATP. The gray bar showed that the total colony count was down to 5% contamination from what had originally been 50%, and there was no MRSA on the hand test culture that was done. So again, this is another study that's a current one that shows the value in the feedback component of monitoring cleaning compliance. And this is the final one that I'm going to show you, and it's ATP compared to marker real-time feedback. Real-time meaning that the sites, and this was done in an ICU, and it used ATP testing, and in this case, the ATP test cutoff was less than or equal to 25 RLUs. So this kit had a much lower cut than the other one I was telling you about. The ICU high-touch sites were all marked with UV visible marker, and in study 1, what they did was even though it was marked with UV visible marker, they went in and they looked at the UV visible marker, and after cleaning and disinfection, if the marker, they assessed it to see if the marker was removed, and if it wasn't moved, it failed, then the failed monitoring test, the staff repeatedly re-cleaned it until it passed using the UV visible marker. And then after that step, they assessed the same area using the ATP level. So you can see what they're doing. They're using one as their method of giving feedback to the staff to repeat a fail. And then after it passes on that one marker, they assess it using the other marker. And then study 2 was where they reversed it. They actually marked all the surfaces with UV visible marker, but this study was assessing ATP as the first assessment. So after cleaning and disinfection, they assessed residual ATP. If it failed, the staff had to come back and repeatedly re-clean it until the site passed. And then they assessed, was the UV marker that had been put on in advance also removed? And the results of this study showed there was no significant difference in terms of the final outcome between each of these modalities. In short, if it failed by UV visible marker and it was re-cleaned until it passed, it will pass the ATP test. If it was assessed by ATP and failures were re-cleaned until they passed, it would pass in UV marker, again indicating that the feedback makes sense and is a valuable thing. Now, the CDC has some wonderful information on its website regarding documentation for cleaning and disinfection monitoring, and there's also some electronic databases that are capable as well. It's important, this particular form from the CDC doesn't include the results of the re-clean and retest, but that should also be done to make sure we track those sites that failed the first time. When they were re-cleaned, did they pass at that point? Okay, so the final poll question that we have is, what method of initial training of the EVS staff at your site is used in your facility? Do you have a dedicated trainer that gauges competency before the new staff are allowed on the ward? Or do any of the EVS staff train on the ward and competency is documented for the new staff? Or are any EVS staff train, they are able to train on the ward, but there's no documentation of competency? So please use your cursor and identify in your facility which of these 3 approaches to training EVS staff is actually used in your facility. And let's have a look. It's always exciting. Wow. This is basically an equal split between all of them. Dedicated trainer gauges competency before allowed on ward, 25%. That obviously is the ideal thing, because then you have a dedicated trainer that makes sure that the staff are properly trained before they're let loose on the ward to do their cleaning and disinfection. And then the other one, EVS staff trains on the ward and competency is documented, is at about 37.5%, and it's an equal split with EVS train on the ward and no documentation. I would, again, this is wonderful data. I would highly encourage those that are not documenting the competency to actually look at the idea of documenting that competency as a minimal step toward this, and highly encourage all sites to use dedicated training and trainers to make sure that there's standardization in the training that's done. Okay, so I hope I have convinced you without monitoring cleaning, you don't know what you don't know, because our data, our own staff basically felt they were cleaning properly 100% of the time. And that's an example of you don't know what you don't know. And so we posed the question, if you look at this toilet, is it clean or not too clean? And monitoring will help you answer that question and make sure that you do know the competency and compliance of your EVS staff and the reduction that helps you in terms of infection transmission. So to summarize the seminar today, environmental contamination with multi-drug resistant organisms can lead to infection transmission, and there's more than enough data to support that. Second point, monitoring cleaning and disinfection compliance is a CSA requirement. And even though it's not a requirement in the CDC documents or the AMI documents, the reality is that is strongly recommended as well. And you can see from the data that I presented, as you start giving feedback, you get better and better compliance with actually the EVS staff doing their job. And there's a pride in them too when they see the data about how well they're doing. And UV visible marker and ATP are key performance indicators that are useful ones to use. So on that note, I will stop and thank you for your attention.
And Dr. Alpha, that was an amazing presentation. I have to say I was like, you're preaching to the choir here about monitoring all these things and telling the staff that you're monitoring them, that knowledge of that, hey, we're going to be opening your sets, or hey, we're going to be looking at the thing that you're doing and monitoring it, and then giving you the weekly updates. I think that's an important part of it too. When they see that you're doing it and then that you give them the updates on it, it really implants it in the staff's mind. And I know that was geared toward EVS, but there's definitely applications into other departments as well, that this is how you provide that feedback for that continuous quality and improvement. So thank you. That was amazing, amazing presentation. I was glued to the screen the entire time. We did get some questions here, and I will go ahead and start giving some of them to you. So when you're using ATP as a measure of cleanliness, could the chemical used to clean interfere with the result or give a false positive?
Yes, there's been both situations documented. We ourselves found that sometimes if there is, bleach component or subsidiaries of bleach components, depending on the formulation, they can interfere. And that's why the manufacturer's instructions for use clearly state that you need to make sure the surface is dried after it has been cleaned before you do the ATP testing. And the reason for that is the interference that's provided by the bleach compounds doesn't interfere once the surface is dried, but it does interfere and can give elevated ATP levels. So it's important to remember, follow the manufacturer's instructions. And the other flip side of it is that the formulations that are used for cleaning and disinfection, it's an important thing to check with the manufacturer as to whether they have any interference one way or the other, whether it's giving you a false ATP signal or whether it's quenching the ATP signal. It's important to make sure that whatever product you're using for cleaning and disinfection, the manufacturer assures you that it's not going to interfere with their ATP test as long as the surface is dried.
Right. Amazing. So here's another one. Why do pre-moistened wipes have a short wet or dwell time, say 2 to 5 minutes, but chemicals applied to cleaning cloths or sprayed have a 10-minute wet or dwell time?
Hey, I'm not the manufacturer of the compounds, but I can tell you my thoughts on it. Okay. The manufacturers state those dry times because they, in their testing, in order to get the kill level for the organisms that they're claiming they have efficacy against, they basically have to identify what the wet time is, and that is a component that users have to be aware of. If your wet time, if it requires 10 minutes of wet time in order to kill, let's say VRE as just an example, that means that you have to keep that surface wet for 10 minutes. Now, we all know that the surface, if you apply once with a wipe of any sort, it won't stay dry for 10 minutes, or it won't be wet for 10 minutes. That means you would have to reapply until you had every surface retaining wetness for 10 minutes in order to get the kill that's claimed by that chemical liquid disinfectant cleaner or disinfectant, whatever component that you're using. And so it's really important to recognize that this is an issue that as you choose the liquid chemical that you're going to use as a cleaner disinfector or as a disinfector, if it's a two-step process, you need to make sure that you consider what the manufacturer tells you is the wet time in order to get the efficacy of killing organisms that they claim. Because it's far better to choose one that's 3 minutes or less, because contact time beyond that is very hard to achieve unless you keep wetting the surface, and nobody wants to do that. It's hard enough to get compliance with going through and getting everything done once because there's so much pressure put on people, in terms of their turnaround times for rooms, etc. So I can't answer the why, but I can tell you that beware and make sure that the agent and the wipe is important too. We compared two wipes. We took pop-up wipes that had the same agent as in the facility I was in, I'm ashamed to admit, but they used to take bed sheets and rip them up into rags, and that's what was used as their cleaning cloth. And we compared that to a pop-up system of wipes. And what we found was that the amount of fluid transferred from our cleaning rags, which were bed sheets, etc., the amount transferred onto the surface was minimal compared to the amount of liquid transferred from the wipe. The wipe transferred more liquid, and the surface stayed wetter longer from the wipe than it did from the cloth that we were using. And yet the cloth system took more chemical overall in a given year and cost more money in many ways than the wipe system. But there are wipes and then there are wipes. So you have to make sure that your wipe is big enough and that it too can keep the system wet for the period of time needed to kill the organisms per the manufacturer's claim. So I'll stop ranting on that point.
No, that's all incredible information, so I really appreciate it. Because you brought up a lot of good points. I mean, in sterile processing, what I have seen people do with the PDI, the super sandy cloth wipes, is they'll do a single wipe down with that cloth, and then they'll assume that it will be a 2-minute wet dwell time, that it was disinfecting that whole wet time. But if you wipe it down and then look at it maybe 10, 20, 30 seconds after, it's completely dry after that point. So there's no wet contact time for that full 2 minutes. I have seen facilities, and I do want to give a shout out to VCU Health down in Virginia. I've seen them actually take those wipes and do the full 2 minutes of wipe down, which is a lot of work, but that's really the only way you get that wet contact time with those wipes. And I know those are a very common and popular wipe. And then the other thing I'll say is that alcohol, 70% isopropyl alcohol, another very popular thing we use in sterile processing, that is very difficult because it has usually a minimum of 5-minute contact time, wet contact time, and alcohol dries immediately. So unless you're really soaking it or consistently spraying it, you're not getting that full contact time. So you have to really look at your label and then really see if you're following what it says to do, because otherwise you're not really getting the kill that you should be getting.
Yeah. And Adam, if there's one thing I can do, the other thing that I forgot to mention during my webinar was the idea of the feedback. Do you do the feedback immediately after you've done the testing on the surface with ATP or UV visible marker, or do you do it kind of on a weekly basis on a team? Ideally, and the data supports this, ideally it's better that if you get a test that fails, you bring the EVS staff back in immediately to actually reclean and disinfect that surface. And so although in our study, remember this was many years ago when there was no cleaning and no feedback whatsoever, and we were giving feedback on a weekly basis, but the more current data would indicate that do give your feedback right away so they come back, clean the surfaces that failed, and and then test them again to make sure they passed. And I apologize, but I forgot to mention that during my webinar.
No, that's incredible information, because I know that's applicable to a lot of different things. It's great for EVS, and I'm sure it's applicable in other scenarios as well. That immediate feedback really is important. And I know as a staff member, I would always appreciate if I made a mistake, I want to know right now. I don't want to know a week from now. Yeah. I'd rather know right in that moment what I did, because it'll be more in my memory, I think. based on what I just did and going into the future. So let's see. And this is, I'm not sure about the wording of this, but I think what it means is if you are using an ATP tester, will viruses show up under the ATP tester?
Okay, that's a great question. I'm a clinical microbiologist, so I love the bug questions. Microorganisms like Staphylococcus aureus, et cetera, have ATP in them. That's their energy source, right? And when we think of viruses do not replicate on their own. They require infecting a human cell, and they replicate inside our human cells. So a naked virus by itself has no ATP in it. So by definition, it could never be detected by an ATP test. But most times when we contaminate the environment with a virus, it's because if we have a cold and we cough and those droplets get on the surface, there's human cells in there as well. And the human cells do have lots of ATP. In fact, there's far more ATP in a single human cell than there is in a single bacteria. And most of the time when we're monitoring cleaning using ATP, we're actually monitoring kind of the organic material as opposed to individual microbes. But in direct answer to that wonderful question, a naked virus by itself, they don't have ATP inside them. But often those viruses, when they're spread in the environment, they come out in patient secretions like fecal material or respiratory secretions, and the human cells will have ATP in them.
Very interesting. I didn't know that about the viruses, so that's good to know. Very interesting stuff. Thank you, Dr. Alpha, for your expertise. We are at time, so I got to get going to the next session. But it was really incredible and really incredible presentation. I think we all learned a lot about the viruses and, of course, that continuous monitoring as well. If you have any questions we weren't able to get to, you can connect with Dr. Alpha via email. Just a reminder, there will be a short 15-minute break before the next session. We're glad you're here, and we'll see you back here shortly.
Thanks, Adam.